Excitation-contraction coupling is when a muscle contraction is sparked by an action potential in the sarcolemma of a muscle cell. In order to contract a muscle, you must first have an action potential which is generated through a neuromuscular junction. The area where the motor neuron meets a skeletal muscle is known as a neuromuscular junction. On the neuromuscular junction, there are both acetylcholine vesicles, represented by the orange dots, and receptors, represented by the green asterisks. Acetylcholine is the neurotransmitter that stimulates receptors and eventually allows the muscle to contract. The first thing that happens within the neuromuscular junction is excitation. During excitation, voltage-gated calcium channels are open, which stimulates exocytosis of acetylcholine vesicles. The vesicles are then released into the synaptic cleft, the blue area between the neuron and skeletal muscle fiber. Once acetylcholine vesicles are released into the synaptic cleft, they bind to the acetylcholine receptors. Binding of acetylcholine to the receptors opens sodium and potassium channels. These channels are ligand-gated, meaning they are activated by a chemical. In this case, that chemical is acetylcholine. Sodium flows into the sarcolemma, represented in the picture by the brown line, and potassium rushes out. The resting membrane potential jumps from -90 to +75, which forms an end plate potential. The change in the voltage of the motor end plate opens the voltage-gated channels in the sarcolemma. This produces an action potential. After excitation comes excitation-contraction coupling. The action potential spread through the sarcolemma enters the nearby T-tubules. This causes calcium gates in the sarcoplasmic reticulum to open. Calcium travels from the terminal cisternae in the sarcoplasmic reticulum into the sarcomere within the muscle fibers. The muscle fibers in this picture are the red areas between the sarcoplasmic reticulum. Sarcomeres are found within the muscle fibers. Found in the sarcolemma are both thick and thin filaments. Actin are the thin filaments, while myosin are the thick filaments. The calcium previously released by the sarcoplasmic reticulum binds to the troponin found on the actin or the thin filaments. This causes the troponin-tropomyosin complex to change shape, exposing the active sites on the actin. Next comes contraction. The first step in the contraction cycle is where myosin heads hydrolyze ATP. The ATP breaks down into ADP, an inorganic phosphate. Myosin heads on the thick filaments bind to actin on the thin filaments, which forms what is called a cross bridge. Myosin heads then perform a power stroke, pulling the thin filaments towards the center of the sarcomere and decreasing the size of the H zone. When ADP binds with inorganic phosphate once again, the cross bridges detach. These four steps form the contraction cycle. If calcium levels are high and more ATP is available, the contraction cycle will continue, and myosin heads will continue in a walking motion. The contraction cycle allows for complete contraction of a muscle. All of these things are done extremely quickly and allow us to use our muscles on a daily basis without even thinking about it.